Everyone, come forward! Come forward! Hello, hi. Good to see you. Wave to the camera. I don't really get together.
All right, cool. Hey everyone, I'm Eleanor, I'm head of DeSci here at SAGE, the SAGE HQ. Thanks very much for coming and super excited to be hosting the DeSciNYC. We're going to be hosting an exciting talk on cellular metabolism, so that'll be coming up very shortly. A little bit about SAGE, so we're in layer 1, fastest EVM parallelized blockchain. We're also dedicated to decentralized science, so as well as other applications in crypto, but also a focus on the application of crypto in the sciences, AI, frontier technology, robotics, etc. We also have a fund, so 65 million in capital, it's called Sapien, dedicated towards building projects on SAE. Yeah, so I mean, that is pretty much everything. I hope you enjoy the talk, and Mike, I'll hand over to you for a couple of notes on DeSciNYC.
Guys, let's give it up for SAE. They gave us this beautiful office, and we're so appreciative for them. And thank you to Eleanor and the entire SAE team. And look at this, we have a koi pond over here. How cool is that? Okay, let's go to the next slide. How do I do it? Who wants a sleep mask? Okay, while we're waiting for the next slide, let's do a sleep mask. A sleep mask. A sleep mask. Oops, sorry. A sleep mask. A sleep mask. All right. In the far back. Sorry. Sorry. Oh, is this? Oh, where's this slide? Okay. 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 Okay, so guys, welcome to DeSciNYC. We host these events like once a month, and you know we have one coming up next time on quantum computing, which is going to be very, very cool. And I'm going to give it to Avinash right here. Yes, he's going to talk about a new type of clinical trial that we're going to be working on.
Thank you, Michael. Hello, everyone. You probably are aware that the microbiome can impact all sorts of aspects of health, but it's still in the very early stages of figuring out how, and most of that focuses on the negatives of how certain strains of bad bacteria can cause problems. But there was recently a lab in MIT that decided to go and look at a bunch of elite athletes and look at their microbiomes and try to figure out which species might be common amongst a lot of elite performers that could potentially lead towards part of their actual success. They were able to isolate a bunch of different strains. Right now, they ran some animal trials, some human trials, they're on the market. I have no actual association with this company in terms of there's no financial link. I just like them, and we're partnering together with Michael to basically take 200 of you, and whoever decides to join, give you free access to 2 different types of these probiotics and figure out at a larger scale, can that impact sleep, digestion, exercise performance? These are all things that have been validated fairly well within the rodent studies and the early human trials, and we're not ones to take anything with small amounts of data and say it absolutely works. So instead we want to get our own larger amounts of data, run our own trials in a decentralized way, and figure out, can we take this and measure its impact on human performance in terms of sleep and physical activity. So if you're interested in getting free access to this fairly expensive probiotic and are willing to do the work to try and test it in yourself and measure the outcomes, come and talk to me or Michael, or there's a QR code somewhere where you can sign up.
Yes.
Excellent. Find us afterwards.
But what's cool about this is DeSciNYC for so long has been just about looking at content and learning about content. But this is one of the first times that a group like this is going to actually be creating new types of science and contributing to the canon of knowledge. So this is going to be very, very cool. You've helped design the study as well. Yeah?
Still
And this I don't know if you know, but he's like an influencer on science, and you should give a talk at some point. I think at some point you will. And maybe you could even talk about the results and how doing it in a decentralized way is possible now.
Yeah, that'd be fun.
Cool. Do we have the link?
Yeah.
Oh, okay. I just want to get the sign up link here.
In case I downplayed it, a lot of people have been reporting really cool results from it, but it's always hard to tell from placebo, so that's why we want to get good data.
Okay, and then we're getting the link up here, and then people that are interested in it. So this is part of DeSciNYC, and it's going to be one of the first trials that's ever been done in a new way. So I'm very excited about this. I don't think that people at crypto and tokens and democratization of science has been done in this way. So very excited. Okay, so without further ado, we are going to bring Timothy on stage. Okay, so let's give a round of applause for Timothy. Timothy is a postdoc at the Rockefeller University studying cellular metabolism. So let's give it up.
Thank you guys very much. I'm super excited to be here and present the work that I've been working on in my postdoc, and I'm starting my own laboratory at Mount Sinai in January. So I'm going to be talking a little bit about the stuff that keeps me up at night, and I'm excited to have a different audience to talk to than a normal academic audience. And I think too, one of the cool things about this, about DeSci and about decentralizing science is finding new revenue streams to fund science, to fund basic science that companies necessarily aren't going to be funding. It's very important research for us to be doing. I hope I can convince you of some of that through my talk and kind of the stuff that I've been working on. Okay, so I will be, so the focus of my talk is really cellular metabolism. And what is metabolism? Metabolism is the sum of all chemical reactions within a biological system. And this is a map showing kind of the central metabolic pathways in mammalian cells. And I know it looks quite complicated, but this is actually a gross oversimplification of metabolism. So we know that there's over 220,000 reported human metabolites in humans, and just in human serum, for example, there's over 4,500 serum metabolites. And so one of the questions that I and so many people for centuries and millennia have been interested in is how is metabolism regulated and controlled, and how do humans and living organisms maintain metabolism to sustain life? And so Aristotle started thinking about this in the 4th century BC, and he made the observation that the food that we eat must be what provides the energy and the building blocks that we need to grow and sustain life in humans. And a couple centuries later, Sanctorius of Padua actually expanded upon this and took very detailed measurements of his consumption and excretion over 30 years and made the observation that for every 8 pounds of food he ate, only 3 pounds of waste was produced. So therefore he said this must be going somewhere. And that principle has really been expanded at the cellular level. So in the 19th century, Ernest Overton studied how metabolites get in and out of cells and how they are transported into the cell and how that enables them to be used for different cellular metabolic pathways and processes. And so really, the key player of cellular metabolism is transporters. And transporters are proteins that sit on the cell membrane that bring nutrients in and out of the cells. And this is a really fundamental process for cellular biology and organismal biology in general. And the fact that this is so important is highlighted by the fact that over 10% of the human genome, all genes that humans encode, they encode for proteins that have transport function. So it's a very key function of cellular homeostasis, but also organismal homeostasis in life. And one of the largest families of these solute carriers is known as the solute carrier superfamily, the SLCs. And there's over 450 of these different transporters, which range in diversity of where they're located within the cell, what they transport, what affinity they have for their substrates. And throughout evolution, these different transporters have evolved to enable organisms and cells to tightly regulate their metabolism. So for example, if we take glucose as an example, which is a very abundant metabolite, there's at least 4 different glucose transporters. And they change in where they are expressed in the human body. So for example, in the brain, you have this transporter called GLUT3, and that has very, very high affinity, meaning it really, really wants to transport glucose. And that makes sense because the brain has a high energetic demands. In other tissues, such as in pancreatic beta cells, the affinity of the transporter GLUT2 has very low affinity for glucose, and that allows it to act as a sensor so that your pancreas can understand whether or not you've recently fed and induced the types of organismal metabolism that your body needs to regulate glucose levels. And so, what do we know about other transporters other than glucose? Well, surprisingly, very little. So of the 456 transporters that I told you about, if we just survey them and ask what is transported by that transporter, almost half of them, we have very little evidence for what it transports. About a quarter of them, we have absolutely no idea whatsoever what it transports. And if we look on the opposite end, where we consider gold standard proof, where we have a structure of the protein bound to its substrate and cellular and biochemical evidence of what it does, that's only 5% of this whole family of transporters. And I told you, this is the biggest and most important family of metabolite transporters in humans. And the fact that we know so little is even more striking when you take into account that over 50% of SLC transporters are associated with human disease. And so when I started my postdoc and some of the work that I'm doing in my laboratory now is, can we systematically identify the physiological substrates of metabolite transporters? And by doing that, understand biology and understand potentially human disease. So we took this GWAS-based approach. So GWAS are genome-wide association studies. So they are studies in which you look at the different SNPs or variants in the genome of people, and then you can associate that with phenotypes or clinical outcomes that those patients have. And so we took this data set for which they had both the genetic information for 6,000 people, but they also measured serum metabolite levels for these patients. And we thought we could use this as a discovery platform to try to identify associations genetically and statistically between metabolites in circulating serum and potential variants in those potential transporters in people, and kind of use human biology as a discovery platform to kind of de-orphanize these transporters. And so normally when people do GWAS, they're looking for genetic variations that are associated with human disease. And what we're trying to do here is the opposite, where we're actually looking for associations in the circulating levels of metabolites of these patients with variants in potential transporters to try to de-orphanize who is transporting what in an unbiased way. And so the idea of looking at differences in serum metabolites to inform physiology is not new. So there are a number of metabolites for which there's human diseases caused by elevated levels of metabolites. So this isn't a foreign concept, but it was a unique approach that we took to de-orphanize these transporters. And we first were able to show that things that you would expect from the literature that you could pick up. So for example, cholesterol is a very important metabolite, and the transporter that takes up cholesterol is LDLR, and we were able to find that variants in this transporter are associated with changes in serum levels of that metabolite. And so we next turned our attention to what was unknown, what's previously unreported. And the one transporter that I'm going to tell you about that I've been working on is this transporter called FOVCR1. It's expressed on the plasma membrane of cells. And we found that it was associated with this metabolite called phosphocholine. And we were interested in this association because it was not only associated with phosphocholine, but also the downstream metabolite of phosphocholine, phosphatidylcholine, which led us to believe that maybe this is a real bona fide hit that has something to do with choline metabolism. Okay, taking a step back, what is choline? I'm sure most of you have probably heard of it before, but it's this 5-carbon essential nutrient that we have to take up from our diet. And it has, once choline goes into the cell, it has multiple metabolic fates of what it can become. So probably most well known is it can become acetylcholine, and acetylcholine is a very important neurotransmitter. It can also be used to make phosphatidylcholine, which I just discussed before, but phosphatidylcholine is the most abundant phospholipid in mammalian membrane. So this is really what makes a cell, this layer of phosphatidylcholine. Additionally, it can be used to make betaine, this other metabolite that's important in 1-carbon metabolism, which plays a role in epigenetic regulation and transcription and other things that I won't touch upon today. But what I want to emphasize is that we know, and I'll walk through this in a second, but we know that choline must be obtained from the diet, and we know that it's a pretty abundant metabolite in human serum. And so the association of choline and physiology really came about in the 1930s. So Frederick Banting and Charles Best were the first to show that if you have dogs that had a pancreatectomy, so they removed their pancreas, they had fatty liver disease, and they found that if you gave these dogs choline, it reversed the fatty liver that these dogs had. Again, showing for the first time that there was an association between the levels of a given metabolite in the diet, choline in this case, and disease in an animal model. And really in the 1970s, before the 1970s, it really wasn't even clear if choline was an essential nutrient for humans. It was thought that animals needed to eat choline, but humans didn't need to eat choline. And plants, for example, can make their own choline. But really that came about in the 1970s, that dogma started to change, and that really came about from studying nursing mothers and the contents of breast milk. So choline is very, very high in breast milk, and choline is passed from mothers through the placenta to developing embryos. And kind of that body of literature in the 90s finally came to the conclusion and recommendation from governmental bodies that choline is in fact an essential human nutrient that you have to take in your diet in order to have normal physiology and homeostasis. And they really observed this for the first time by doing studies in the 80s and 90s where they gave people choline deficient diets and then asked what happened to these people. And these people end up developing fatty liver disease, they have muscle wasting, and they have liver wasting, the liver degeneration. But one of the coolest things about those studies is that when you reintroduce choline into the diet of people who you've taken choline away from, those phenotypes and those diseases all go away. Again, showing this tight link between the nutrients that you take up in your diet and your health outcomes. Okay, so we know that choline, because of its structure, must be transported into cells. It cannot just passively diffuse across the membrane and get into cells. And going back to kind of the association studies that I showed you before, we hypothesized that maybe this transporter that I discovered, FOVCR1, is a transporter of choline and allows choline to get into cells. Okay, now getting into some actual science. How do we show this? So we can take cells and genetically engineer them, so we knock out a gene and then add it back with an expression system. And we did this in cells, and then we just did metabolomics, where we can look at all metabolites detected in the cell and ask what goes up, what goes down. And when we did that, we found that in cells that don't have FOVCR1, this transporter, they have very low levels of phosphocholine, this choline-derived metabolite, as well as choline and another choline-derived metabolite, betaine. And there's another approach that we can use, especially in metabolomics, which is using carbon tracing. So we can take advantage of the fact that some carbons are naturally occurring to have, they're heavy, naturally occurring, so we can feed these heavy carbons in a metabolite, such as choline, to cells, and then trace how those carbons move through the metabolism and ask where did that carbon go when we fed it to the cells. And so when we do that, we see that cells without FOVCR1 cannot incorporate choline into the downstream metabolites of choline metabolism, like phosphocholine and betaine. Okay, now to really demonstrate that FOVCR1 was a choline transporter. We used radioactive choline, and again, similar types of experiments gave it to cells with and without FOVCR1. And the hypothesis being that if FOVCR1 transports choline, you should need FOVCR1 to see a radioactive signal of choline. And that's in fact what we saw. So in the orange you see here, we saw both time and dose-dependent uptake of choline through the transporter FOVCR1. And really, the gold standard in terms of metabolite transport is showing that in a liposome system, so a liposome is a cell-free system, it's a fake membrane, circular membrane in which you put in your protein of interest. And so without any other components, just your transporter of interest, do you see uptake of that metabolite? And that's in fact what we do see with FOVCR1. So FOVCR1 alone can transport choline into liposomes. We also then did some work doing the cryo-EM structure, where we resolved a structure of FOVCR1 bound to its substrate, choline. So I'm showing you here too, this protein exists in these 6 hetero pseudobundles that rock back and forth to transport choline. And this is just zooming in on the pocket where we found choline bound to the protein. And there's these specific residues or specific parts of the protein which bind and stabilize choline so that it can be transported. And when we mutate some of these residues, specifically this tryptophan W125, we can completely block choline transport. So again, we've really kind of nailed down from a biochemical point of view, this is the substrate of FOVCR1, it's choline. Okay, so this is all well and good, like you found a transporter, it transports something. Do cells even care about having this? And surprisingly, not really. So only when we really, really deplete choline do we see a difference in cells' ability to grow in culture conditions. In normal conditions, these cells grow completely fine. And if you think about it, I just told you choline is super important, so how is that the case? So we used, that's a question. So how do cells sustain choline metabolism and proliferate without FOVCR1? And we used CRISPR-based genetic screens to uncover some possible ways that the cells are using to code this. And this is technically a little challenging, but conceptually very simple. You take cells with and without your gene of interest, and then you just grow them, and we use this pooled genetic CRISPR system that each cell has a different gene knocked out in it. And then over time, in a pooled fashion, you ask if that gene that got knocked out is essential for cells in your condition, not having FOVCR1, for example, to grow, then at the end of your experiment, you should see that cells in which you've knocked out that gene are no longer there. And you can do these types of experiments, and it's the power of unbiased genetic screens. And what we're able to find is that, I don't want to walk through the details very carefully, but we're able to find that there were other ways that cells were able to compensate for not having choline transported through FOVCR1. So one of them is the salvage pathway. So cells can actually take up lipids extracellularly, which contain choline, and they can take that up and break it down and recycle it. So we're able to find out new biology in this respect. And also, we were able to see that one of the hits in our screen was this other transporter, which is a family member of FOVCR1, called FOVCR2, and we were able to show that that itself also can transport choline. Okay. Again, this is cool, this is in mice, but what happens, or this is in cells, but what happens in an organismal context? What happens if you don't have FOVCR1 and no choline transport in mice, for example? What happens? Well, consistent with the idea that it's essential, mice without FOVCR1 don't make it to birth. So they end up dying at embryonic stage E12.5 in mice. So you can see there's the wild type mice, or the mice that only have one copy of FOVCR1 gone, completely fine, they're born, they're normal. The ones that have both copies of this gone, developmentally have defects. You can see that you don't have to be an embryologist to see this. The head is different size. You see the eyes in the wild type and the heterozygous that you don't see in the knockouts, and even the size of the embryo is different. So this really shows that this transporter is actually necessary for mammalian development. And when we did metabolomics on these embryos, we found that the knockout embryos do in fact have decreased levels of choline and these downstream metabolites. Again, showing that the problem with these is probably that they don't have enough choline for their downstream metabolic functions. And then we tried to ask, could we actually rescue these mice that don't have FOVCR1 by giving supraphysiological amounts of choline, so very, very high doses of choline to the pregnant mothers during pregnancy to try to extend the lifespan of these knockout embryos. And what we found is that we could significantly extend the lifespan of these knockout embryos. So for example, in the control arm, knockout embryos at day E15.5, we don't see any of them. I told you they die at E12.5, that's 3 days earlier, you don't see any of them. But the knockout ones, when we give really high levels of choline, we can still see live embryos at this time point. Again, there's still a lot of developmental problems with them, so we're not able to fully rescue it, but I think this is very strong proof in mice that the thing that these mice are feeling, these embryos during development, is that they don't have enough choline. Okay, so why is FOVCR1 loss embryonically lethal? The truth is we still don't have a clear answer for that, but I want to show a little bit of data from this. And what we think is happening is that the mice that don't have FOVCR1 during development, their mitochondria, which you guys probably know, the powerhouse of the cell, this important metabolic organelle, is very messed up and dysfunctional in the knockout embryos. And you can see, I think, just appreciating through these EM images, what the mitochondria look like. They're dark and dense. The darkness comes from the electrons in them. And they have these beautiful structures of this double membrane bound structure. And you can see that in the knockout embryos on the furthest right, you have the cristae, which is the folds of the mitochondria are messed up, they're swollen, and we know from other data that I'm not showing here that they're not actually behaving properly. And what we think is happening is that the phosphatidylcholine, one of the lipids that I talked about at the beginning, is a really important lipid for mitochondria, and perhaps by restricting choline through FOVCR1 not being there, they're not able to have enough phosphatidylcholine to make effective mitochondria, and maybe that's why they're dying. And we're working on this to try to figure that out. Okay, what about in humans? And I think the beauty of basic science is that if you find something truly fundamental to basic science and biology, it usually is implicated in some type of human disease. And that's the case that we found as well. So they're actually rare, but there are dozens of people that have variants in FOVCR1 that causes this ultra-rare disease called PCARP disease. And that stands for posterior columnar ataxia with retinitis pigmentosa. It's a long name, but it's a debilitating disease in which they have degeneration of the retina, so they eventually become blind. And they also develop problems walking and ambulating because they have degeneration of their spinal cord. And before our work, they had made the association that this variants in this gene were associated with this disease, but what this gene transported and what potentially was the etiology of the pathophysiology of this disease was not clear. And also, this other transporter that we discovered in our genetic screens, the cousin of FOVCR1, FOVCR2, is also associated with this less rare human disease called Fowler's syndrome. And kind of the point of this work is that you could hypothesize that given that these patients have variants in these transporters, and I think we have pretty convincing evidence that what they're missing is choline, you can imagine devising very easy dietary interventions to help these patients by giving them more choline. And that's work that we're doing right now with clinicians to try to understand, or try to help these patients. But also we're trying to understand too, choline itself exists in this form furthest on the left, but there's other metabolites, as I told you about in the genetic screens, from which metabolite cells are very thrifty in the ways that they can regenerate choline. And really the important part they need is this part that's highlighted in yellow. So we're playing around with different formulations of different choline-derived metabolites that we can give these patients to try to provide them with more choline so that they can have a better expectancy in terms of lifespan, but also in terms of the pathologies that they're suffering from and the disease that they have. And finally, one of the things that I'm interested in as a basic biologist is that when we identify what a transporter does, that gives us a genetic handle that we can then go ask very fundamental biological questions. So now that we know that FOVCR1 is a choline transporter, we can go knock it out in very specific cell types and ask, what is the role of choline in the nervous system in mice? And when we knock it out, what happens in these mice? And that's kind of the work that I'm doing now. It's really going full circle and really understanding new biology based on very fundamental de-orphanization of these metabolic transporters. And I want to highlight this is just one of these 450 transporters. There's a ton of work to do to de-orphanize these, but by understanding how nutrients get in and out of cells and organisms, we can start to build a better understanding of how cellular metabolism really works, and it can point us into directions of potential therapies for patients that have variants in these transporters. And with that, I want to thank my lab and all the people that I work with, and I'd be happy to take any questions. Thank you guys so much for listening.
All right, does anyone have any questions? Yeah. Take the mic so the people in virtual video land can hear. Here, I'll work with it.
Awesome. Thanks so much for the talk. It's like Alzheimer's, other dementias, or disease of acetylcholine deficit. So you got like anticholinesterase inhibitors like Aricept, long-standing drugs. Just curious, like, research or maybe I guess research you've done into that class of disease and FLVCR1 dynamics there.
No, this doesn't work. Thank you. That's a very good question. And that's exactly what we're doing now. So we have knockouts of these mice that have some very Alzheimer's neurodegenerative-like symptoms when we've made new knockout animals of these. So that's exactly what we're trying to do and try to understand the interplay of if choline is really necessary through FLVCR1 for the function of making acetylcholine. And in these patients, for example, would cholinesterase inhibitors, as you suggested, be beneficial? And I think they would. So that's where we're ongoing and working on. Yeah. Thanks for the question.
So in the example you were showing earlier, when you had the knockout mice, that they were able to go and inject larger amounts of choline to sort of overcome that. How does that translate into the amount that the human body can actually handle? Like if you're trying to think about in humans for those two diseases that you discussed, what's the upper limit on choline intake that would be able to potentially exist and not cause other problems? And how does that balance happen? And also, if you're talking about recycling mechanisms that could potentially go and be the alternative ways of making use of the choline, you said that's the way. The modifications of the choline molecule are the ways that you are going to try and get, versions of choline that can actually work for people who have these sorts of knockouts. Are there particular ones that you have found so far in these knockout mice that are looking to be more promising?
Yeah. No, that's a great question. So first off, so in terms of the amount of supplementation that we're able to do in mice, this is not necessarily the most directly translatable. So so we're giving these mice dietary amounts of choline, but I think that the limit for that is the intestinal absorption. So if you can eat bucket loads of choline, it's very hard to overdose in terms of eating choline. The biggest side effect is that you smell fishy because you make this trimethylamine, your gut microbiome actually breaks down the choline. So what we're doing in these mice is actually injecting IP choline. And so I wouldn't suggest doing that in patients because if you inject too much choline, you can cause a cholinergic crisis in the mice, for example, seize and die. So we found like the absolute level that the pregnant mice can take, and that's what we were using. So in terms of the therapeutic benefit, I think as he suggested over here, maybe acetylcholinesterase inhibitors would be a good approach. So for people who don't know, acetylcholine is a neurotransmitter. It gets released in the synapse between neurons. And normally there's this protein called acetylcholinesterase that breaks down acetylcholine into choline and acetate. And it's the same principle as like an SSRI, for example, that increases the level of serotonin in the synapse. By blocking the breakdown of acetylcholine, you're increasing the amount in the synapse. So that would be a therapeutic benefit that you could go through. And then like you suggested about the other forms of choline, potentially some of those would have less of a cholinergic effect if you were to give intravenous amounts. So I know, for example, I've seen like Gwyneth Paltrow says she takes lysophosphatidyl in the IV drips. So potentially you could think about ways of doing other types of choline-containing molecules or metabolites that would be less averse in the patients that have the variants in FOBCR1 and 2. But it's hard. We still don't even know how choline is absorbed in the intestine. So we know that you have to get, I think for women it's like 425 milligrams per day. Men it's like 550 milligrams. But we don't even know what the main dietary source of choline is. Like we know, for example, that it's in fish, meat, spinach, eggs, things like that. But we don't actually know, is that coming from free choline that's in those, or is it coming from choline-containing metabolites like lysophosphatidyl and is the absorption that's happening in the gut of lysophosphatidyl or is that being broken down into choline and then being taken up into the gut? So there's it's a very important metabolite, but there's very fundamental things that we don't even know how it gets into circulation. But that's the kind of thing we're playing around with.
And how long does it take, let's say, hypothetically speaking, if there was a timing for recovering the system, let's say the mice, for example, how long did it take you to make it back to normal level after you tried the steroid thing? Number one. And number two, what was the threshold and what would be the, again, hypothetically speaking, what would be the threshold for a human? You said 550 for male, 425 for women. If you wanted to compensate more for it, yes, of course, we have to evaluate how low is it, and then take it back. But let's say you're recovering from a disease or something like that that happened. Let's say you went down to 50%. Just again, hypothetically speaking, how long would it really take it to, let's say, we'll take it up and we'll leave it there, and is it like 2 months, 3 months, or have you tried something like that?
So the mouse experiments that I've shown so far, we're doing we're treating them from conception. So they've always had elevated levels of choline. The amount that we inject into the mice is like equivalent to what a human needs. So it's very, very high amounts. And the thing I want to emphasize too is in cells that don't have FOBCR1, they cannot take up free choline. So I think the way that we're getting choline into these cells is there's just so much outside of the cell that some of it's like leaking into the cell. So that's the other thing is like, even if you were to make the choline level back to what a normal patient had that has a variant with FOBCR1, they still can't take up that choline. Do you know what I mean? Because choline, so the
cell is actually damaged.
The cell is not able to take up that free choline. So a better approach would maybe be using a different choline-containing metabolite because it's probably a different transporter for that metabolite. So patients that have variants in FOBCR1 may be okay at taking up glycerylphosphatidyl. So if you give them that, you're still giving them the same molecular toolkit that they can then work with to do their metabolism, but you're circumventing the problem they have in getting into the cell, if that makes sense.
And in terms of timing?
I would think the problem with any neurodegenerative disease is like, once the horse has left the stable, it's a little late. So it would be preventative would be the best case scenario. If you were to screen people that had variants in this, the more you can give them excess amounts of choline, the better. Because it's very hard to, once neurons have degenerated, remake those neurons. They're gone. So that's what I would say about that. And it would have to be probably a forever thing, I would think.
Thank you for the talk, found it very interesting. Curious about the funding from Merck. So what's the end goal of the research? Does it lead to something which is translational? What are the applications in industry?
Yeah, so the funding that I have from Merck is just they fund a certain number of postdoctoral fellows at Rockefeller. So I have that funding for a year. So it's not even like project-based. So there are ways, of course, that pharma companies will work with investigators, but that wasn't really the case for this. In terms of the therapeutic benefit, I think these would be considered ultra rare diseases that I don't think many pharma companies would touch this type of work. The one good thing is when you're working with metabolites and nutrient supplements, a lot of that is not considered a drug, right? You could tell a patient that you should go get nature-made choline and try that. You don't even need to file an IND or to do those types of trials. So that would be a way that we could help these patients without going through typical pharmaceutical trials. But I think big picture, understanding how choline is taken up and what that plays a role in different diseases, going to Alzheimer's or something like that, could be something where a pharmaceutical company would be interested. And I can say, for example, prostate cancer, they use choline PET scans to stage prostate cancer because prostate cancer really takes up a lot of choline. So like, potentially, if you had an inhibitor that blocked FOVCR1, that really you have found a therapeutic window that hit cancer cells but not normal cells, that would be a good therapy. We haven't talked to any pharma companies about that kind of stuff, but that's kind of what we're thinking. So hopefully that answers your question.
Thank you. Any more questions?
So two quick questions. So you mentioned, for example, choline can be absorbed into multiple ways in the body. So how does the body know which way to kind of, which would be the best use? And two, is there a way to kind of, for neurodegenerative diseases, to screen for whether the problem is, for example, it's like the inability to absorb certain metabolic
Yeah, it's a good question. So I would say that probably there's no, in terms of the association of the problems in absorption and disease like neurodegeneration, I doubt there's probably genetic defects in transport in patients that cause common diseases like neurodegeneration. It's possible that deficiencies in diet could be associated. So they have done studies in mice, for example, that if you manipulate the amount of choline that they have in their diet, and there's a mouse model that gets Alzheimer's already, they get Alzheimer's even worse and even earlier. So that's a possibility. And then in terms of your first question, like, very interesting, how does, it's not just for choline, for any metabolite, how does the cell know what it should go to make and how does it do that? The way it usually works for most of these metabolic pathways is it's kind of a funnel. So it comes in and it has multiple routes of where it can go. And the way that the cell usually changes the rate or the flux from which it goes down this path or this path is the amount of the enzymes that do the work to make it go down this side or down that. So example, you would shut down the enzyme that makes this so that in this cell type, you're not making the metabolite that you didn't want to make. But how, it's a very good broad question. How do cells sense and know what metabolites they have? How do they regulate that? That's a wide open field and something we and other people are very interested in.
One last question.
Yeah.
Just a quick question. In the HET case where they have one copy, what's happening there? Is it like half the cells are getting it, half are not?
No,
or
so
yeah.
the HET would be all cells have one copy of FLVCR1. So they're not, it's not a mosaic where some are knockout, some are wild type. It's all of them have one copy. But what you can tell from that is that having one copy of FLVCR1 is sufficient to do the job. And that actually is what we see in some patients that we've been working with, where their parent has one copy gone, and the other parent has another copy gone in a different place, and only when they're together does their child have a problem. So having half is enough. But that's not the case for every gene, but in this case it is.
Got it. And how does it render like on the cell membrane? Like, do they have like half as many transporters?
Usually, but it depends. Some that's also controlled at the level of transcription. So some genes you don't need to have both copies to have the same number of protein. And it could be that the cell, we don't know this is speculation, it could be that the cell senses that they don't have enough of it and they tell the cell to make more of that. And there's a compensatory mechanism that maybe they have the same amount.
Cool, thanks.
How many questions are left? You?
Wait, so are you proposing for prostate cancer they knock out the gene?
No, I wouldn't propose to knock it out, but in theory, like a lot of these drugs would be to inhibit the function of it. So if you could find something that could stick in the middle of the transporter and block it so that it can't do that, can't take up choline, that would be
So you would be suggesting to take up less choline for prostate cancer?
That would be the idea.
I see. But wouldn't that come with other side effects?
Of course,
like
that's every drug.
ADHD?
That's
Yeah,
every
I see.
drug. So it's always looking for a therapeutic window in which having that drug would hit the cells that you want to hit and cause an effect there without causing some other toxicity. But that's with every drug.
Did the drug company show interest in that?
We've never pitched it to them,
Okay.
and I've never really, I did cancer in my PhD, planning to do more of it in the future, but that's not something it's just kind of throwing it out there. As an
Okay,
idea.
I see.
But something that would be interesting, but you'd have to find a molecule that can inhibit it.
Yeah.
And I would be a little concerned, given the phenotypes that we see in the mice, that the therapeutic window we have to hit cancer but not hit any other cells might be too tight. It's not even worth doing that.
Okay, I see.
Okay, the next question.
Just quickly, the first sort of thought that I think probably most people have when they see the GWAS with the metabolites thing is, wow, that's a lot of hits, and then you chose one, and this was really great talk, like tremendous amount of work though. So are there any sort of thoughts or like initiatives to sort of parallelize this process?
Yeah, so for example, the other work that I didn't present now, we published all the associations that we found. And so one of the things that I think it goes back to DeSci is decentralizing and putting back out there. And I think my whole career, I hope to have a long career, I can probably do, I don't know, tens of these, but I can't do hundreds of these.
Right.
So it's a big open project. And again, you have to choose what you're interested in. And the way we did it is like, for me, it's like if it's evolutionary conserved, that's more interesting for me. If there's patients that have variants in this, that have a disease, that's interesting to me. But again, whenever you do these large types of fishing expeditions,
Yeah.
how you choose what to go after is always difficult. But I think one of the important things and the push that there is, I think, in the scientific community is to make all that data accessible. So maybe some investigator really, really cares about glutamine or some other metabolite, they can go find associations and they can work on that.
Cool. Thank you. All right. So got you a big science grade again hat for your wonderful talk. And thank you again to Say, and thank you. We have more pizza and drink. Feel free to chat and mingle. So thank you all for coming. Grab some pizzas. If anyone needs the QR code for the study, let me know. I don't want to interrupt, but I was ready to talk to you.
That is Talking to the wrong people. Anyone else wish to talk to me?
That was like 25, 26, and then we're at 8 years later.
I worked in that lab a long time ago. Still a third lab. Was it on the first feed already? Right? Or was there another reaction? Yeah, we've been at this office for a very long time, and the lab is for us to figure out. Thank you so much. It's a wonderful thing.
Hi, thanks you to everyone who joined us. This was an amazing event. We are wrapping up, so if you guys could just head to the exit. Elevators and staircases are right ahead. Bathrooms are all along the left wall. Thank you guys again.
Hey, Mike, can you want help? Oh, good. Yeah. I think we're good. I mean, do you get some
We would, I'm here today. I didn't need to be like, I meant to help me move these chairs back to this conference room. That was the main objective in pulling this table to the front. That'd be done.
Perfect.
That'd be it.
Okay, yeah.
Assuming this is walls. Yep. Just gonna conference room to the format. Last time. Alright. Cheers. Cheers. Yeah, you guys know.
Is it all? Oh, so that's not
Thank you.